Yeah, our today's topic is cell division. So first we will discuss what is cell division. So cell division is the process by which parent cells parent cells produce their daughter cells. Daughter cells. So cell division is a process by which the parent cells produce their daughter cells. So every cell which is capable of division has to pass through a cell cycle. Every cell which is capable of division has to pass through a cell cycle. Then only it can undergo mitosis or meiosis. So what is this cell cycle? This cell cycle is a sequence, it is a series of events, which was first worked out by Howard and Pillay in 1950. So Howard and Pillay, they for the first time worked out the sequence of events which leads to cell division. So this sequence of events is known as cell cycle. So how we define a cell cycle? So cell cycle is the period from the end of one cell division to the end of next cell division. So this period, which starts from the end of one cell division and terminates in the end of next cell division, this period is known as cell cycle. So it is the period from the end of one cell division to the end of next cell division. So this intervening period is known as cell cycle. Or we may also say it like this: cell cycle is the period which starts from the formation of a new cell, its growth, metabolism, and then its division into daughter cells. Daughter cells. So this may be another definition of the cell cycle. So it is the period which starts from the formation of a new cell. Then this newly formed cell, it grows in size. It assumes the size of the parent cell. Then it carries out all its metabolic activities. Every cell is supposed to do some function in our body. So this newly formed cell, it grows in size. Then it carries out all its metabolic activities, its functions. And finally, this cell also divides into two daughter cells or daughter cells. This cell finally divides into daughter cells. So this period from the formation of the new cell till it grows, till it carries out its metabolic activities, and till it divides into daughter cells. This period is known as the cell cycle. And this cell cycle was worked out first by Howard and Pillay in 1953. So every cell which is capable of division has to pass through these sequences of events, has to pass through this cell cycle. We divide this cell cycle into following phases. Cell cycle is divided into following phases. cell cycle is divided into following phases. The first phase of cell cycle is known as interphase. Second phase of cell cycle is known as M phase. And the third phase of cell cycle is known as cytokinetic phase. So a cell cycle is divided into following phases. First phase is known as interphase, second phase is known as M phase, and the third phase is known as cytokinetic phase. This interphase, it is the longest phase of the cell cycle, and it is a non-dividing phase. It is the longest phase, and it is a non-dividing phase. In this phase, the cell doesn't divide, so it is non-dividing phase. Some authors misinterpret as arresting phase. It is not arresting phase. Although the cell does not divide in this phase, but it is not the resting phase because the cells are metabolically most active in this phase. So it is a non-dividing phase, but metabolically most active. It is metabolically most active phase. It is metabolically most active phase. Now we further divide this interphase into three more phases. It is first divided into G1 phase, S phase, and G2 phase. G1 phase, it is also known as first growth phase. First growth phase, or it is known as gap first phase. Gap first phase, right? S phase is known as synthesis phase. Synthesis phase. Because in this phase there is synthesis of new DNA. And G2 phase, it is known as second growth phase. Second growth phase or gap second phase. Why we call it as gap? Because between one cell division and the next cell division, there is first gap, right? Interphase is the gap between two cell divisions. So the first gap is in this phase, second gap is in this phase. That is why we call this as G1 phase or the first growth phase or the first gap phase. This is the second growth phase or the G2 phase or the second gap phase. And S phase is the synthesis phase. Now we have to discuss what happens during these phases. We will discuss it one by one. So first we will see what happens during the first growth phase. What happens during the first growth phase? G1 phase. What are the main events of this phase, G1 phase? In this first growth phase, cell grows in size. When the parent cell divides into daughter cells, this newly formed daughter cell, it is very small in size. So it has to enlarge. So it undergoes growth. So that is why we call it as first growth phase. So in this phase, the cell grows in size. So the cell size enlarges, right? In this phase, cell carries out synthesis of new cell organelles. When the parent cell divides into daughter cells, parent cell distributes certain cell organelles equally among the two daughter cells. For example, mitochondria. For example, plastids. These mitochondria, their number is then restored. When the daughter cell is produced, this daughter cell restores first the number of mitochondria. In the parent cell, suppose there were 8 mitochondria. When this parent cell divides, it will distribute 4 mitochondria to this cell and 4 mitochondria to this cell, right? So the first growth phase, in this phase, cell synthesizes these new cell organelles, right? In which it is deficient, right? Then cell carries out synthesis of various cellular molecules. Cellular molecules. At the time of cell division, the cytoplasm of parent cell is equally distributed among two daughter cells. So the cytoplasm gets reduced. Cytoplasmic contents get reduced. So in the first growth phase, cell carries out synthesis of cytoplasm. It carries out synthesis of proteins. It carries out synthesis of lipids. It carries out synthesis of carbohydrates. It carries out synthesis of cytoplasm, right? So it increases its cytoplasmic contents so that the cell is self-sufficient in all the cytoplasmic contents. So in this phase, cell carries out synthesis of various cellular molecules, like it forms proteins, it forms some lipids, it forms some enzymes, it synthesizes RNA, it synthesizes other materials. So in the first growth phase, the cell enlarges in size, cell synthesizes those cell organelles in which it is deficient. Then cell carries out synthesis of protein, synthesis of lipid, synthesis of enzyme, synthesis of RNA, and synthesis of other biomolecules, so that this cell has all the required elements in it, right? So this leads to the growth of the cell. When the cytoplasmic contents increase, the cell grows in size, right? And in this phase, the cell also carries out synthesis of enzymes, which are required for the second phase, that is the replication of DNA. In S phase, there is synthesis of new DNA. Their DNA will be formed. So all the enzymes which are required by the cell for the replication of DNA, those enzymes are formed in this phase, in the first growth phase, right? So before the cell enters into the S phase, a checkpoint operates here. That checkpoint is known as a restriction checkpoint. Before the cell will proceed into S phase, it has to pass through a checkpoint. That checkpoint is known as a restriction checkpoint. A restriction checkpoint. At this restriction checkpoint, a series of enzymes is produced. CDK, transcription factors are activated. What is this CDK? Cyclin dependent kinase transcription factors are activated. So they activate all the necessary enzymes which are required in S phase. So cell will only proceed into S phase when it has all the necessary materials for going into S phase. So a series of enzymes is activated. Cyclin dependent kinase transcription factors, they become active and they activate all the enzymes which are required in the S phase. So cell has to pass through this restriction checkpoint. Any cell which is deficient in the necessary enzymes which will be required in the S phase will not be allowed to go into S phase. So for going from G1 phase to S phase, a cell has to pass this restriction checkpoint, or we call it as G1 checkpoint. So for going from G1 phase to S phase, a cell has to pass this restriction checkpoint or G1 checkpoint. At this point, a cell must have all the necessary enzymes which are required in the process of DNA replication. If this cell possesses all these enzymes, if these enzymes are all activated in G1 phase, then only the cell will go from G1 phase to S phase. So this at the end of this G1 phase, a cell has to pass through this restriction check point. So it is ensured whether the cell is ready to go into S phase or not. If the cell is not ready to go into S phase, then it is stopped. Then it is not allowed to enter into S phase. When all the required material, when all the required chemicals, enzymes, proteins, etc. are present in the G1 phase of the cell, then only the cell is allowed to enter into S phase. Now we will summarize what happens in the G1 phase again, right? So in the G1 phase, first the cell shows growth. Cell enlarges in size. So how cell enlarges in size? This cell, it carries out synthesis of its various cellular molecules. Proteins are synthesized, enzymes are synthesized, lipids are synthesized, other cellular molecules, they are synthesized. Cell also carries out the synthesis of deficient cell organelles. When the parent cell has divided into daughter cells, some of the cell organelles are equally distributed into two daughter cells. So the daughter cell is lacking half of those cell organelles. So first cell, this cell first carries out the synthesis of those cell organelles. So all the cell organelles are formed. All the cell organelles are made ready in this G1 phase, right? As the cell enlarges, as it accumulates new cytoplasm, then this cell will go into S phase, right? But before going into S phase, it has to pass through a checkpoint known as restriction checkpoint. At this restriction checkpoint, a series of enzymes is activated. There is a series of enzymes known as cyclin dependent kinase transcription factors. They ensure that cell has all the necessary enzymes which are required in DNA replication. If cell is having all these necessary enzymes which are required in DNA replication in S phase, then only cell will be allowed to go from G1 phase into S phase. If the cell is deficient in those enzymes, then the cell is not allowed to go into S phase. That is why we call it as restriction checkpoint, because the cell may be restricted from entering from G1 phase into S phase. If the cell is completely ready, then the cell will be allowed to go from G1 phase into S phase, right? So these are the changes which occur in the G1 phase or the first growth phase of the cell cycle, right? Now we will see what happens in the S phase. The second phase is known as the S phase. We will see what happens there in the S phase. Now S phase. This S phase is known as synthesis phase. Why we call it as S phase, synthesis phase? Because in this phase there is synthesis of synthesis of new DNA. New DNA. That is why we call it as synthesis phase. So how DNA is synthesized, new DNA is synthesized in this phase? We all know that DNA replication is a semi-conservative process. So a parent DNA molecule starts unwinding, and on the two strands of the parent DNA molecule, new DNA starts being formed, right? So DNA replicates a single DNA molecule, one DNA molecule. It replicates. It replicates. How it replicates? What is the process? I'm not supposed to discuss that, right? So one DNA molecule replicates into two DNA molecules. Two DNA molecules. So from a single DNA molecule, now two DNA molecules will be produced. This will be our one DNA molecule, and from here another DNA molecule, right? So this is the parent DNA. So in the newly formed DNA, there will be a parental strand and a new strand. Similarly here, there will be a parental strand and a new strand. You all know DNA replication is a semi-conservative process. Semi-conservative process. So one parental strand is conserved, and on that parental strand, a new strand is formed. So on this parental strand, a new DNA strand will be formed. On this parental strand, a new DNA strand will be formed. So from a single DNA molecule, two DNA molecules are produced. So this happens in the S phase of the cell cycle, synthesis phase. In this, there is replication of DNA. So a single DNA molecule which is contained in a chromosome replicates and it forms two DNA molecules. So before the onset of S phase, this chromosome, it was having a single DNA molecule. Now as it entered into S phase, this single DNA molecule replicated and two DNA molecules were now produced. And now these two DNA molecules, they are contained in the two chromatids of the chromosome. So in the S phase, the DNA content of the cell, in the S phase, DNA content of the cell is doubled, but the chromosome number remains same. So in S phase, DNA content of the cell is doubled, but chromosome number remains same. So suppose this cell was having one chromosome when it entered into the S phase. In the S phase, it is DNA replicated. This single DNA molecule, it produces two DNA molecules. One DNA molecule and another DNA molecule. Both the DNA molecules, they are still contained in the single chromosome. Before entering into S phase, in the G1 phase, this chromosome, it had a single chromatid. This chromosome had a single chromatid. Now when it enters into S phase, DNA replicates and this newly formed DNA molecule, it is contained in separate chromatid, right? Now this chromosome, it has got two sister chromatids. So chromosome number of the cell remains same. But DNA content of the cell is doubled. Right? So in S phase, there is synthesis of new DNA from a single DNA molecule, which is contained in a single chromosome, right? Another DNA molecule is formed. So both the DNA molecules, they are still present in the same chromosome. In the beginning, this chromosome was having only one chromatid. Now as the DNA replication occurs, now it develops a second chromatid. Now each chromosome has got two sister chromatids containing two DNA molecules in them, right? So in S phase, there is synthesis of new DNA. So DNA content of the cell is doubled, but the chromosome number remains same, right? So this is the chromosome with double DNA. This is the chromosome with single DNA molecule. In this chromosome, there are two DNA molecules. So chromosome number of the cell will remain same. In case of human beings, it will be still 46, right? After DNA replication, in addition to DNA replication, centrosomes of the cell also replicate. Centrosomes also replicate in S phase. There is replication of centrosomes as well in this S phase. So to summarize the events of S phase, so we now know that in S phase there is replication of DNA from a single parent DNA molecule. Two new DNA molecules are formed, right? The parent DNA molecule was contained in the single chromatid of the chromosome. Now, as it gives rise to two DNA molecules, now they will be contained in the two sister chromatids of the same chromosome. So every chromosome has got now double DNA content. So the chromosome content of the cell remains same. Chromosome number of the cell remains same, but the genetic content, DNA content of the cell gets doubled. In addition to this replication of DNA in S phase, there is there is replication of centrosomes as well. So centrosomes of the cell, they also replicate in the S phase. Then from S phase, the cell goes into G2 phase. Now from S phase, the cell goes into G2 phase. G2 phase is the second growth. G2 phase is the second growth phase. Now second growth phase. Or we call it simply as G2 phase. So what happens in the second growth phase or G2 phase? The cell further enlarges in size. So cell shows growth further. Cell shows further growth. That is why we call it second growth phase. Cell makes synthesis of new proteins, enzymes, RNAs, and other biomolecules, which will be required by the cell during the process of this cell division, during the M phase. So in second growth phase, the cell further enlarges in size. It carries out the synthesis of all the materials which will be required in the M phase of the cell division, in the mitotic or meiotic phase of the cell division. So cell ensures that it has got all necessary proteins, it has got all necessary enzymes, it has got all RNAs, right? It has got all the biomolecules which will be required in the this mitotic or meiotic phase. So simply we say cell further grows in size, it carries out synthesis of new cytoplasm and all the materials which are required for going into this M phase or mitotic phase or meiotic phase. So all those materials must be ready with the cell, right? Before the cell goes from second growth phase into M phase, it has to pass another checkpoint. That is known as G2 checkpoint. G2 checkpoint. Or we call it as DNA damage checkpoint. DNA damage checkpoint. So the cell is now getting ready for entering into mitotic phase or meiotic phase. So before it goes into mitotic phase or meiotic phase, it is ensured that cell is having normal DNA content in it. During the process of replication, there was no damage caused to the DNA, right? So a series of enzymes is activated. Cyclin dependent cell division cycle 2 gene. It is activated. Cyclin dependent cell division cycle gene 2 is activated. So before the cell goes from G2 phase into M phase, it has to pass through this G2 checkpoint or DNA damage checkpoint. So at this checkpoint, another series of enzymes is activated. Cell cycle independent cell division cycle gene 2 is activated. It checks whether the DNA of the cell. It activates the enzymes which check whether the DNA of the cell is normal or it has suffered any damage during the process of replication. If these enzymes detect that any damage has been caused to DNA during the process of replication, so that damage is immediately repaired. So DNA is repaired before it enters into mitotic phase. However, if the cell is not able to repair the damage caused to DNA, then this cell will not be allowed to go into M phase, mitotic phase or meiotic phase, right? So before the cell enters into mitotic phase or meiotic phase, it has to pass through this G2 checkpoint or DNA damage checkpoint. At this checkpoint, it is ensured that in the process of replication, no damage was caused to the DNA, right? So a series of enzymes is activated. Cyclin-dependent cell division cycle gene 2 is activated. It stimulates some enzymes to check for the DNA damage. When the cell is ready for entering into M phase, mitotic phase, then the mitotic phase starts. If the cell has suffered any DNA damage, either that damage should be repaired first or the cell is stopped from entering into the mitotic phase, right? So that is all about the three phases of interphase. So in the cell cycle, we had the first phase, interphase. We divided that interphase into G1 phase, S phase, and G2 phase. And we discussed the sequence of events which occurs in those different phases. I think this is sufficient for today. Thank you.